This lesson is about the cell cycle. You'll be able to describe and sketch a diagram of the cell cycle, state the key events of each stage of the cell cycle, and outline how cell cycle control is facilitated by cyclins and cyclin-dependent kinases. So first, the cell cycle is often diagrammed on a circle like this, kind of like a pie chart, and we're going to divide it into 4 unequal size pieces, and we're going to label those M phase, G1, S phase, and G2. Now let's look at what each of these mean and what happens during each phase. The M phase is the mitotic phase. And a little bit confusingly, the M phase consists of both mitosis and cytokinesis. Now remember that cytokinesis is not properly part of mitosis, but it is part of the M phase. So if progression is going clockwise, as it is meant to be on a diagram like this, then the M phase consists of 2 parts. First we would have mitosis, and then after that we would have cytokinesis. So the M phase consists of mitosis and cytokinesis, as we learned about in a previous lesson. We also talked in a previous lesson about how replication of DNA doesn't happen in mitosis. The nucleus enters mitosis with all its chromosomes in a 2-chromatid state, made from 1-chromatid chromosomes elsewhere in the cell cycle. And that happens in S phase. S stands for synthesis, and S phase is where DNA synthesis occurs, where 1-chromatid chromosomes make 2-chromatid chromosomes. So S phase is DNA synthesis. What about G1 and G2? G1 and G2 phases are where the cell grows and also where it replicates its organelles. Replication of organelles doesn't happen alongside replication of DNA, but rather it happens in G1 and G2. By the way, G stands for gap. There's also another stage that the cell can enter from G1. It's not shown here, and that's called G0 phase. And G0 phase is a non-dividing phase where the cells are just carrying out their functions, whatever kind of cell they are. They're performing their function in the body. And as I've shown here, cells can leave G1 and go into G0 and then come back into G1. Now it's important that a cell's progression through the cell cycle be very carefully controlled to ensure that it's only dividing when it's ready to and healthy and not otherwise, because uncontrolled division leads to disease like cancer. So let's look briefly at one of the ways that it does this. First, a bit of background. One way that protein machinery can be switched on or off is by adding or removing phosphates to the protein. That can activate or deactivate it. And an enzyme that adds phosphates is called a kinase, whereas one that removes phosphates is called a phosphatase. So there are enzymes called cyclin-dependent kinases, abbreviated CDKs, that are present throughout the cell cycle. But they're not active unless they are bound by molecules called cyclins. So imagine that you've got a cyclin-dependent kinase, something like this, let's say, and its function is to activate other proteins by adding phosphates to them. And it's not switched on unless it's bound by its partner cyclin. So this is a cyclin, and this is a cyclin-dependent kinase, and right now it's on, and then it can add phosphate groups to other proteins, switching them on. Cyclin-dependent kinases, the bottom part, they are present all the time, but the cyclins themselves are fluctuating up and down throughout the cell cycle. Cyclin levels fluctuate up and down cyclically in the cell cycle. Whoops! Should only say cell once. Now, together, therefore, cyclins and their partner cyclin-dependent kinases drive progression through the cell cycle through this process of adding phosphates. Now, at certain checkpoints in the cell cycle, the cell takes stock and determines whether or not internal and external conditions are adequate for it to be safe for it to continue progressing. For example, it might check, has all of its DNA been replicated correctly? Is the cell attached to an external environment, etc.? There are a number of conditions that it checks to make sure that it's safe to progress. And so these checkpoints exist in different places in the cell cycle And if those conditions aren't met, the cell won't progress. Now, there are also other control pathways that intersect with and influence the cyclin-cyclin-dependent kinase pathways at these points too. So this is just a first look at cell cycle control. In the next lesson, we'll look at what happens when cell cycle control goes wrong and we end up with cancer.